Hello, everyone. Thank you for giving the opportunity to present our work that we have been working lately close to 1 year. So I'm Rajiv Sharma and my colleague CJ Young. So we co-hosted this presentation together. So this is a little bit different than what has been up to now presented by another colleague, or let's say another presenter. So what I'm going to present in the next few minutes is new ways into plant breeding using eigen-GWAS or env-GWAS and origin-specific genomic selection. This is so the first part of eigen-GWAS and env-GWAS is going to be covered by me, another one by my colleague CJ. So we are located in SRUC. This is a Scotland Rural College, located in one of the campuses in Edinburgh, quite near to the Edinburgh University, and that is majorly involved in crop improvement, agriculture, animal breeding in the rural sector of Scotland. So I have structured my slides according to this slide. So first I'm going to present what exactly by mean of when I say an env-GWAS or var-GWAS, what exactly I'm going to present in the next slides. And then just a case of plant breeding as its success story over the years, because plant breeding is mainly been started, let's say 50 years back. And can we use this as an environmental trait? So that's basically the major idea of the whole presentation that I have been conducting today, or I have been presenting today. So in the next slides, I will present these in little detail. And so I'm going to present two major examples. So these are coming from wheat and barley on an env-GWAS and also on the environment GWAS. And at the very end, I will conclude on that. So 4 data sets are going to be presented in the next few minutes. And this includes UK winter wheat, Brazilian spring wheat, UK winter barley, and UK spring barley. So in general, most of the crop plants, they've gone through breeding and registration of varieties. And this in majorly is structured according to every year. So the breeders are actually putting in some of their breeding lines, and they've gone through 1 or 2 year testing. And if they got selected based on their performance, then they go on to the next stage, that's the national list. And if they are selected, then they are going to go and recommended for the farmers in the recommended list. So you could see here, this is a kind of a cutthroat situation where every year breeders are actually putting in a lot of breeding material, but only few of them gone through that kind of curtailing process. And each step is the place where the lines got dropped out just because of their non-performance or because of not performing well with their other counterparts. So at the end, only few lists can make it through, and this is the one that gone to that recommended list. So this process of plant breeding is happening quite long, from let's say 1990, 2020. And you could imagine that the line that, for example, is been recommended in a year somewhere in the 1950 is probably present for a few years until it got disappeared because it's not in the recommended list anymore. And similarly, some lines, they can still be there because of the market demand. So now the question is, can we use this as an env-GWAS? I mean, the age of the variety, can we use this as an env-GWAS trait? And this is what we try to use, because selection has been happening on those lines when they've gone through that national list and recommended list. So another introduction of that is basically over the years, there is a genetic gain on the yield. And this has been shown by a 2011 paper of Ian McKay, where he showed that over the years across the 3 crops, this is wheat, barley, and brassica, you could see that there is a linear trend of increase in grain yield or over the years. And if you use just the year as a proxy for the yield, you could very well somehow predict the value of the yield in the next years if you just put it on that linear graph. So like I said to you, so I have used here env-GWAS of age. So here age of in this case is being used, and what you can see here is the GWAS of the wheat, 21 chromosomes of wheat on the y-axis Manhattan plot, and on the top is the winter wheat varieties, and the down is the another set comes through the Brazilian spring wheat varieties. And what you can see here is when I use here the env-GWAS age as a trait, I could pick up not random things, but actually peaks on chromosomes. That, for example, in these cases, 8 regions could be picked up with high significance. I mean, I can't see in these cases similar trend being picked up in two different plant material. For example, the winter wheat varieties and the Brazilian spring wheat varieties, they pick up completely different regions. For example, the one that I have highlighted here. Similarly, the age is being used for spring barley and the winter barley analysis from UK. And what you can see here is that we could bang on pick up the peaks on PPDH1, for example. This is a flowering time gene on chromosome 2H, and SDW1, this is a height gene on chromosome 3H. And similarly, we also picked up other interesting region. One of the most interesting region in this case is chromosome 5H short arm peak that I have highlighted here. Why I'm trying to say that, because this region is being continuously picked up across the winter and spring barley, irrespective of their growth habit. Then we thought we could just combine the data set together and see whether within the, irrespective of the growth habit, whether when you combine the spring and the winter barley together, whether we still pick up that region or not. So what you can see here is, again, we pick up that region on chromosome 3H across 103 genotypes. This is even more interesting even after putting in the growth habit as a covariate. So nothing actually changed. So on the top, you could see the growth habit as a covariate used for GWAS analysis. On the down is without covariate, nothing changed except few magnitude differences in significance values of one or the other. And that suggests that the peak that we picked up with the age as env-GWAS on chromosome 5H is quite promising. And of course, like I explained here, so age could be used, and I have shown here that we could pick up the regions that underlie the genes of flowering time and height. But what is interesting here is, can we get more by using the trait data per se? So in order to validate those, we use the, for example, the national trial data. So on the down is the GWAS using the national trial yield data with treated and untreated on the down here, and on the top is the env-GWAS. So you can see here, I have used here the first year, last year, and the lifespan. So this kind of duration between the first year and the last year has been used also as env-GWAS. So, and across these 5 GWAS analyses, you could see the 5H region is quite promisingly present across the whole peaks, suggesting that this region that we picked up by using age as a trait is quite promising. And furthermore, I don't have time to show it, but we know that this has something to do with disease resistance, because you could see here that the untreated yield data from the national trial is not much significant comparative to the treated one. The treated and untreated means in the treated yield data across the national trial, fungicide and other pesticides are sprayed on the plants on the other hand, untreated is without the treatment of that infection like pesticides. In order to visualize, like I said to you, so normally such kind of analysis are possible when you have two different populations and that you can separate out and you can use basically the conventional approaches like FST. But in these cases, we pick up the regions that is kind of quantitative. Just to show you an example, here is a picture of one of the pig on chromosome 2H at 20 centimorgan, quite close to PPDH1 gene, flowering time gene. So we have devised this as a, we called it a rocket plot. What you can see here is the two colors that are the codes of the alleles. One shown here with blue color, another one shown with red color. So the modern allele that says starts quite late, but is been quite significantly enriched over the years. And now, if you look at the varieties of the modern time, they are mostly of modern type, not the olden type. On the other hand, you can see that somewhere in the 2000 and 2010, these old alleles were still present, although in a low frequency. So what I'm trying to say here is that this type of analysis is only able to pick the region that we could show with the age as a trait. Similar to that, we also use the eigen-GWAS to on the 4 data set that I have shown here. So 10 major PC principal components are used in this analysis. And from the inner circle to outside, I have shown here the GWAS of the eigen-GWAS. So 10 PCs from inner to the top. So it's difficult to visualize much, but what I'm trying you to show is these major things. What is interesting in this case is that wheat analysis of eigen-GWAS pick up quite congruently peaks like 1B1 or introgression on chromosome 1B that I have shown here. This is also present in Brazilian spring wheat. And in addition to that, we also pick up, for example, 5B and 6B region that I have highlighted here across the two datasets. Contrary to that, in UK Winter's eigen-GWAS analysis, we could not see anything congruently present across the two datasets of winter and spring type. On the other hand, you could see large peaks individually pick up with one or the other species. And if you delve deeper into that, you could see that these are actually being explained by flowering time or dwarfing gene that probably has been selected in those genotypes. So in conclusion, I'm trying in the last slides to show you that env-GWAS and environment GWAS could be used. And I have shown also with the subset of yield data from the national and recommended list that one could pick up the regions and the age data as a proxy for the yield. And because we actually pick up in a subset the same region that we pick up age as a proxy. And also on the eigen-GWAS analysis, it's a bit different story. So we pick up identical region across the two data sets of wheat, Brazilian wheat, and UK wheat, although they are geographically quite separate from each other. And on the other hand, the barley eigen-GWAS doesn't show anything congruently present across two data sets. And finally, I want to acknowledge the SRUC team. This is Professor Ian Mackey, Wayne Powell, and CJ, and also the other collaborators who have given us access to all the resources that have shown here. Thank you very much for listening.
Great, thanks very much, Rajiv. Let me move over to CJ. We did have a question come in, but we will have those do those questions at the end. So let me just switch over to CJ now. There we go. Okay, CJ. Okay, I will see you all.
Great. Okay. Okay, hi everyone. Thank you for tuning in, and thank you for the lovely introduction by Joran and Rajiv. And thank you for the opportunities for me to speak to you guys today. So as Rajiv has presented the first part of our joint talk series, so I'll do the second part, which is on a flavor of genomic selection called origin-specific genomic selections, in short, OSGS. So as we know is that there's a lot of limitation in genetic diversity in modern crop. And that is in particular due to breeding process where we basically have a lot of bottlenecks and selection and drift going on over the years that give rise to what we see in our modern crop materials. And so then what makes it interesting for people researching nowadays in crop breeding is that we want to be able to bring back the diversity. And a lot of it has to do with going back into the landrace or wild relatives as a source of genetic diversity. So one such example of the program would be like the Seed of Discovery program being run at CIMMYT, where they're bringing in landrace, introducing the novel variations into their pre-breeding program. And so here we came up with a concept of genomic selection that we call origin-specific genomic selections, and that has to do with that depends on the parents that we're considering here. So here we look at the biparental population. So you have an elite parent and an exotic parent. And what these are basically the elite would just be the modern crop variety. An exotic line could be a landrace, could be a wild relatives or something else. And so what happened next is that once you make a cross, you can apply genomic selection to try to create a new elite variety that would have favorable alleles for your trait of interest. So because the way it works in selection is that it's a matter of probability. So in this case, it's not surprising that the elite parent would contribute more favorable allele than the exotic parents. Take, for example, yields. Modern variety are well adapted to high yielding and stuff, but your wild variety are not. So in that sense, it's not surprising to find more favorable allele coming from the elite parents. And so as a consequence, when you select out from these kind of populations, you end up picking a variety that is highly similar to the initial elite variety. And what we attempt here to do is kind of a cheat that we want to separate out the pool that we pick from in the selection process. So on top here, we separate out the favorable elite alleles, and the bottom, we get the favorable exotic alleles. So now we have these two pools in separation. We can apply different amount of selection force on each of them to create our desired new elite variety. So in this case, we're able to drive more favorable allele from the exotic parent. So this is kind of the concept of it. In reality, how do we achieve that? It's kind of fairly simple. I'm going to skip over the technical stuff, so I'm going to try to do them in a fairly simplified way. But first, what we start with is when you have your genomic marker, so in the form of A, C, T, and G, what we need to do is just to first code it according to parent of origin. So in this case, what I'm coding is -1 for homozygous elite parent, +1 for homozygous exotic parent. You could switch this around too, and it would still be the same. It's just a matter of sign that you need to keep track of. And of course, the heterozygous class, we need to keep it right in the middle, and it has to be 0. So then one sign would confer to one parent versus the other. And next is that we can put this in model, in a mixed model, to estimate marker effects. And it doesn't really matter which model you pick, as long as it's your favorite models like R-blob or any other Bayesian model. So what this does is that you can get estimate of the marker effect kind of similar to what you get out from GWAS. You have marker 1 giving -1 effect, marker 2 giving -1, and so on. So in this case, I'm just illustrating it in a simple case of 5 markers. So since I coded the elite parent as -1, any of the allele with the effect, a negative effect, would be considered favorable elite allele. So in this case, I've coded them red. And similarly, the green, these two are the favorable exotic allele coming from the exotic parents. So next, how we're going to do is that we can predict the favorable parental contribution from elite or exotic. And here we have 2 examples here, both of which have 4 favorable allele in total and 1 unfavorable allele. So in this case, the total score for these 2 lines would just be 3. But when you separate them out based on the elite or exotic contribution, the first line has less elite contribution than the second line, but it has more exotic contribution than the other line. So if you're just going through a standard genomic selection protocol, you wouldn't tell these 2 apart because they're the same. But if you're actually going deep into the origins where this contribution is coming from, you will probably want to select the first line because you're gaining more new alleles coming from the exotic parents. So that's kind of a schematic of how it works. And so to demonstrate that, we analyze a set of data using the HEB25 barley num population. This is basically made of an elite variety Barkey crossed against 25 wild barley. And so to make things simple, I'm showing you U in one of the barley family as an example here. And what we did is that we start off with estimating the marker effect using RR-blob. So as you can see here, I'm plotting out the marker effect along all 7 chromosomes. And anything that is positive represents the exotic favorable alleles. And anything that is negative represents the elite favorable alleles. And from here, and also when you put them in a histogram, you can clearly see that there's an unequal distribution of favorable alleles. So in this case, it's quite clear that it's biased towards the elite parents, where 57% of the alleles are coming from the elite parents, in contrast to just 43 from the exotic parents. And so from there, we can use this different partition to make predictions, such that you can use the standard way, which is all marker, or you can split them into the elite or the exotic and make prediction out from it. But then the question comes is that how do we actually use the prediction? And so next is that before I go into the prediction, I just want to highlight again that, across all the 25 NAM family, you see a large disproportionate distribution of favorable allele in elite parents, especially in family tree. Almost 3/4 of the alleles are coming from the elite parent, not the exotic. And so then what happened next is that we want to be able to select based on these 2 partitions. And so what we do is that we can do some simulation exercise. So here we have a, here we have a simulation population with 60% favorable elite allele and 40% favorable exotic allele. And so what happened here in the top panel is that we look at the breeding value over 5 generations of selections. And here I coded blue as selection using OSGS and orange as selection using GS or genomic selection. So as you can see, over time, the breeding value increases, which is good. And it's not surprising that OSGS is actually lower than GS in terms of breeding value, but I'll come to it later on at the bottom. But it's kind of tailing along the over the generation. So next is that this second panel I'm showing is the proportion of favorable elite alleles that is present in the population over the same generation of selection. So what you can see is that genomic selection is really good at increasing the proportion of favorable elite alleles. As you can see here, it increases by almost 50% over 5 generations, while the exotic allele kind of, while OSGS kind of keeps it at a slow increase pace. And on the other hand, when you look at the proportion of the exotic alleles, which is the one that we're interested in bringing into our breeding population, genomic selection is pretty good at driving it to a low level, such that you end up with about 50% of what you originally had in your initial populations, while OSGS is able to keep it at the same level throughout the selection process. So this is kind of important because it gives us a way to keep the exotic allele in our pre-breeding populations, so we can actually be more efficient and effective in bringing in novel allele into our breeding program. And next is that we want to be able to further evaluate OSGS to GS. And here, what we did is that we continue on with one generation of simulation using the previously analyzed barley num as an example. So in this case, you can have many different selection weights on OSGS. As I mentioned earlier, you have the elite and the exotic pool. So if the selection weight is 0, you'll be basically selecting simply from the exotic pool. And if the weight is 1, you're selecting based on just the elite pool. And anything in between is just a varying degree of selection weight. And so obviously, these two extremes are bad because you don't exactly want to just go for these two, because which I'll show in a bit that why they're bad. But anyway, so then what we compare is that we compare the performance of OSGS against genomic selections. And here I'm highlighting the example with the selection weight of 0.5. So in this case, what it means is we're giving equal opportunities for the exotic and the elite alleles to be selected. And so what we see here is that the breeding value after selection is fairly similar between OSGS and GS, it's just slightly lower. Similarly, the proportion of favorable elite allele in this second panel are slightly lower in OSGS, which is not surprising because we're giving less preference towards the elite selection in this selection weight. And next is that we're able to increase the proportion of the exotic allele, and this is with the selection weight of 0.5. And what happened next is that when we have a range of selection weights, obviously, as I said earlier, the extreme would be bad because you end up with really poor population after selection. But in general, what you can see here is that since the goal is to bring in novel alleles from exotic parents, anything that increases the proportion of exotic allele is good. So as you can see here, this is what I call as a more desirable. And at the same time, if we can minimize the loss in elite allele and the breeding value, that would be the most desirable. So in this case, for this specific example using yield in barley, a selection weight somewhere between 0.4 to 0.6 would be plenty good in bringing in the exotic allele. And so it comes to a final remark is that, I hope that I got the message to everyone, since I notice it's quite a broad audience, is that, we have a simple and robust approach to partition the favorable parental contributions that we can use directly in a pre-breeding program to bring in more exotic alleles in a very efficient way. And obviously, this is very straightforward, and it works really well with other pre-breeding approaches that have been suggested previously in how to bring in exotic alleles. And of course, what we have demonstrated here is just all using a single trait, but it is possible to extend our concept into multi-parental populations such as MAGIC, or even multi-traits, where you're considering bringing in a new variety from a different environment that have certain quality that you're interested in, but maybe it doesn't have as good as a yield as your elite variety. So there's a lot of kind of a road that we can go down from here. But again, I hope the message is clear that we are able to partition the favorable parental contribution. Yes. And with that, I would like to thank all the people who have worked on this project, including Rajiv, Gregor, and Sarah, Wayne, and Ian. And if you guys have any question, you can check out the paper, the link below, or you can contact me later if you can't think of any question right now. So with that, thank you very much.
Thanks so much, CJ. And if anyone wants that paper, I put a link to it in the chat box of the webinar software there. So check it out if you'd like. So we have a few questions. So Rajiv, if you want to come back up as well, a couple of questions that you guys can answer. Let me first ask CJ though about this new technique, the OSGS technique. So can you give us an idea about what the benefit is of bringing in these exotic alleles rather than the elite alleles? Can you give us like a concrete example, a real life example where that would be beneficial?
So it has to do with basically crop improvement. I'm trying to use a more quantitative trait like yield as an example rather than a disease resistance. But I guess disease resistance would be a better example to describe here. So basically, for example, is that you have gene A that confer disease resistance in your modern populations. One is being overthrown, you kind of need to look for new things. And so that is, you start looking into these landrace and wild materials. But of course, when you're talking about disease resistance, this tends to be qualitative traits, and you don't exactly need to go into genomic selection type of stuff to get that in. But it's kind of a better example to demonstrate than a quantitative trait. So that makes sense.
Yeah, absolutely. It just brings in new alleles that would be lost by the conventional analysis, I guess. Yeah. Okay. So we have a couple of questions from Lydia Sierra Gonzalez, and first one came in when Rajiv was talking, and she asks, how do you decide the threshold to use when you run the GWAS programs?
I mean, in general, there are multiple ways to do the threshold. So the one of them is the QQ plots, and then make the decision based on that. Another way of that is just look at number of, so it's kind of a Bonferroni threshold, number of the tests that you get. So it is kind of counterproductive in the sense, because if you have million of SNPs, then you are basically gonna get a very high threshold that normally not even a single marker will pass through it. So by and large, you have to make a kind of a pragmatic decision. So in general, I mean, this is just for the comparative purpose. I put 4 as a threshold that is quite high in general for quantitative traits, and I mean that way I could at least show the congruency across the. But of course, I mean, there are multiple ways to do that threshold value.
Okay. So another question from Lydia. She asks also, how will the new GWAS approach work with traits that have low heritability?
I mean, In this case, what I have tried to show here is the env-GWAS of age as a variety. So age is in general, how you could explain it as a heritable trait is a bit difficult in that sense. But in general, the age of the variety is kind of been gone through national trial type of screening, where only the best could survive through that kind of window that I have tried to depict with that cartoon. So in general, of course, the env-GWAS has not got any trait per se, but I have tried to show it using the yield trait that the peaks that I have been picking with the env-GWAS of age as a trait could be confirmed by the yield of the national trial in that particular case that has not very low heritability but also high heritability in that particular case. But of course, I mean, in general, is a difficult question if you have low heritable trait.
Okay, I see. Okay, so I'll encourage other people if you want to ask a final question. Let me just ask CJ. So just to clarify, so with the new technique that you mentioned, so can you go back to any previous data set and reanalyze it using your new technique to reveal new alleles, or do you need new data to be able to do that?
Yes, technically applicable to any kind of data set. The only important matter is to be able to identify the parent of origins. So it all depends on whether you have the data, the marker data for the parents or not. So.
Okay, I see. Okay, so we haven't got any other questions coming in, so I think we will leave it there. Thanks very much for you guys for presenting.